You will be convinced, I think, by the end of the hour that Paul is something of a force of nature in science. He began his training at that great institution for giving people a leg up in science and other fields, the City College of New York, finished his PhD at the University of British Columbia, and then spent 20 years working at the Brookhaven National Laboratories. In the 1990s, he went to Rutgers, where he has been the last 20 years, and is the Board of Governors professor of, I think, practically everything. He's a professor of business and economics and geology and biochemistry. I'm not sure he's a professor of history, but he's probably working on it. Paul is one of the few people I know who is comfortable working on scales of time and space that go from the kind of femtoscale phenomena of photochemistry to thinking about the nitrogen cycle across all of Earth history. He has made notable contributions to our understanding of the carbon cycle, life in the oceans, particularly photosynthetic life in the oceans. His group has been working more recently on everything from the genomics of calcium carbonate skeleton deposition in corals to the actual detailed protein structures of the proteins that are involved in transferring electrons in metabolism. You've already heard about some of Paul's prizes. I won't mention many of the others, except to note that he is a member of the National Academy of Sciences, among other things. He is also an accomplished author. His book on aquatic photosynthesis with John Raven is, I think, the best book available to learn how we know what we know about photosynthesis. Paul and I together actually edited a book on the evolution of photosynthetic life in the oceans. And recently, and I'm not paid to say this, Paul published a really good, accessible book called Life's Engines, How Microbes Make the Earth Habitable. And I think this is the kind of thing we're going to hear about tonight. If you're intrigued by the presentation, I do recommend this book. It's terrific. And so without further ado, Paul Falkowski.
Thank you very much, Andy. Thank you very much, Andy, and thanks for inviting me here. It's a pleasure. So I just want to begin this by thinking about, whoops. Thinking about a bridge. In this particular case, it's an obvious bridge. And if you think about evolution, you know where we've come to, but you don't know where we began. So origins of life is one of the most challenging problems facing science. Actually, as my friend and colleague Nick Lane says, it's the black hole of science. It's an embarrassment. And it's a very complicated problem. Obviously, in this university, Jack Szostak and others work on this from one angle. I'm trying to understand it from another angle. But we know a certain amount about how we have a certain number of ingredients. So this is a very, very famous plot, and I'm not going to bother you with all the details, but I just want to give you a little bit of an idea. Victor Goldschmidt, who created this plot originally, was a Swiss Jew born in Zurich, and his father took a position at the university in Oslo. So when he was a young man, he actually graduated university, I believe at the age of 21 with a PhD. He became a professor himself, and he created a field which is geochemistry. He created it. And he was obsessed with understanding the distribution of elements in the universe. So he did several things. He analyzed meteorites, he looked at spectra of stars, and so on. And we come up with this basic plot, which is the two first elements are hydrogen and helium, which were created about 13.8, 13.9 billion years ago in the Big Bang. And everything else on this plot, the astronomers call metals, but everything else is created in a supernova, not our star. a very hot, short-lived star. And I put this up for you because if you think about them, these elements, there are two ends of the spectrum here. There's hydrogen and helium. This is the fusion reactions, which creates a star. So that's an energy source by fusion. And then within our planet, we have uranium and thorium and one other element which is not visible here as itself it is, potassium-40, which is radioactive, which by fission reactions create a heat engine within the planet. So those are the two major driving forces of life on Earth, both from radioactivity from two different processes at opposite ends of the spectrum of distribution of these natural elements. And now we can go and walk across. There's hydrogen, helium, then we come to a lithium, beryllium, boron hole, and then there's carbon, nitrogen, oxygen. We can walk and walk and walk, and there's phosphorus and sulfur. So those 6 elements make up life. Sometimes they're rearranged. We just take the C and put it in front of the H, and we call it schnapps. So that's how a student can easily remember what the elements are. All right. So these elements form the proteins in your body, the fats, the carbohydrates, and the nucleic acids. Those 6. So I just gave you the ingredients. How did we get the proteins? How did we get the nucleic acids? We don't really know. And I've been fascinated by that problem for a long time. So if you are a young student in this audience and you think we know stuff, we don't know a lot of stuff. And that's good for you. Now, I just want to make this comment because it's really important to discuss what life does. And life is electric. It's not a metaphor, it's a real thing. So you're breathing right now. It doesn't feel painful, I hope. So as you're breathing, you're taking in oxygen from the atmosphere of the room, which is not made in the room, right? It's coming from the outside world. And you're breathing out 2 gases. One is obvious to you, is carbon dioxide, but the other is water. You've actually put a hydrogen, 2 hydrogen atoms onto an oxygen to make water. That's called reduction. I'll explain it in a bit. So you've oxidized, you've made energy, you can get the energy from food. You're oxidizing the food. So you're moving electrons around. So all organisms derive energy by moving an electron. And all substrates and products must ultimately be recycled. So now you create a bug that makes a substrate, and a bug, it produces a product, and that product is a food for another bug. And so you create, as I'll show you, an electrical circuit. So life is really an electrical process. In the Archean oceans, the source of the electrons and hydrogen was hydrogen gas itself. There's no hydrogen gas in Earth's atmosphere anymore. We still get a little bit of hydrogen gas coming out of deep-sea hydrothermal vents, but for all intents and purposes, hydrogen itself is not produced on the surface of the planet, as abiologically. Iron. Iron 2 is a soluble form of iron when there's no oxygen. It was quite abundant in the early oceans. Hydrogen sulfide, that's a rotten egg smelling gas. We produce this all the time under anaerobic conditions. If you go down to the beach and you see some mud and you dig it up, you probably get that smell. And then sugars. Today, by far, the most prevalent source of electrons and protons, hydrogen itself, to make sugars, is liquid water. So here's a glass of water. The light is shining on it. Nothing's happening. The only thing that will happen to that water is it'll evaporate if I leave it there long enough. But if I put a photosynthetic organism like a cyanobacteria in there, it will split with this light energy that water into oxygen and electrons and protons. That's called a catalyst. So on a planetary scale, oxygenic photosynthesis takes water and carbon dioxide, we mix them together in the presence of light, and you produce a sugar, candy. This is candy. It doesn't look like candy to you? And this gas which you're breathing, right? Now, if you think about this for a minute, you're breathing the gas, somebody else made the gas, you can't make this gas. You're doing this, you're taking the sugar, you're combining it with oxygen, and you're producing these two gases, water and carbon dioxide. Now, if there's oxygen in the atmosphere, it means somebody didn't respire it. So you had to bury the candy. You have to take it away from the children. You have to put it somewhere in the back of the cabinet so they can't find it. That lockbox is the Earth itself. It's the lithosphere. It is geology. Now let's take a look at this planet. We call it, and Carl Sagan called it the pale blue dot. This is a real photograph. I think we actually did go to the moon. It was not fake, okay? Now, if you look at the planet, it's blue because we have this liquid water on the surface of it. We're the only planet remaining in our solar system that has liquid water on the surface. The only planet. Now, if you think about that, let's just, again, for this younger people or people that are more interested in things that they don't know, we don't really know where the water came from, believe it or not. Now, it was imported early in Earth's history. It probably mostly came from meteorites. And that's the source of the surface water. And if I took all that water on the surface, it looks like a huge, huge, huge amount. But it's spread very, very thin. 4,000 meters is the average depth of the ocean. If I take all of that water and I put it into one big ball, that's the volume of the ocean. It's very small. It's amazing that the liquid water has remained on the surface of this planet for over 4 billion years. Mars had liquid water on it. No more. We don't know about Venus. Now, I'm going to switch gears for 1 second here. This is a very, very famous sketch. Darwin was interested in the origins. What was the bridge? What was the other half of the bridge? Where did it come from? Right? That's the origin of species idea. And he wrote a doodle, this little doodle. It's in Notebook B. It was never published like this. There's only one illustration in Origin of Species, and it's a minor variation of this, which is much more detailed. So when the book was published on November 24th, 1859, John Murray and Sons put this one illustration in the book. The rest of the book is just words. And the idea here is that one organism, through variation and selection, which he didn't really understand in 1837 when he wrote this little doodle, led to variations in species that led to variations in species. So you have an origin and the observed distribution of organisms. This is the original tree of life, the original literal tree of life. It was a doodle. I wish I could doodle like that. So this is what a modern-looking tree is. And here is where I think we had a disconnect between the microbiologists of the day and the people that studied plants and animals. And the disconnect was Darwin could see variations in animals or plants, and he could understand processes that would select for them and give from an origin a different shape or form or phenotype. He didn't think about microbes. And I'll get to that in a second. Virtually everything down here, not virtually, everything down here is a microbe. Here are land plants. I mean, there are people that study land plants. I don't know why. These are animals. Animals are very closely related to fungi, which is quite obvious if you really look at some behaviors. But virtually everything else here, again, is a microbe. The vast, vast, vast majority of life on this planet is microbial. Now, in order to see microbes, you need an instrument. You can't see them with your naked eye. You can see manifestations of them, but you can't see the individual. You can't follow behaviors. You can't look at shapes and forms. In 1865, Robert Hooke, who was then an original fellow of the Royal Society, had constructed for him a microscope which had two lenses. It was a very, very bad lens system. For the time, it was the best they could do with grinding. But at most he could magnify was about 20-fold. With this, he cut a very thin piece of cork with what he called a knife as keen as a razor. And he looked at the distribution of the structures that he saw in this cork. And he noticed there were little boxes. And they reminded him of the cells of monks. So he called them cells. This is where we got the word. Now, the cell is the smallest unit of self-replicating life that we have on Earth. If I take an organelle out of your body like a mitochondria, it does not replicate itself. If I take any other organelle and I take a piece of a cell, it does not self-replicate. The cell is the unit of self-replication. So let's talk about the discovery of cells. This is a microscope that was designed by Leeuwenhoek in Delft. Now, Leeuwenhoek was a textile merchant. He was quite wealthy. And he became interested in the number of threads that would be in a textile. So he designed a very simple lensing system, which was very clever. He pulled a piece of glass, he would break it in thin thread, and then he'd put it back into the flame, and that would form a little ball, almost a perfect sphere. After it cooled, he would polish it. He never gave away how it was polished, and even after he died. Then he would mount that little ball in a little bit of a hole up here, and he created these little devices. Now, the device is actually about this big. I was at the Royal Society a few years ago when I was writing the book, and I wanted to see, I understood that there were 4 original Leeuwenhoek microscopes in the Royal Society. So I asked the librarian, could I see one of the original Leeuwenhoek microscopes? He very ceremoniously comes back with white gloves, gives me the white gloves. I have to put the white gloves on. He opens up a white box. Inside is a piece of silver containing a silver object like this. And he says, be very careful with this. He says, this is the only one we have, but it's not original. I said, what happened? He said, well, members of the society borrowed the original ones and they didn't return them. Okay. Anyway, the way this worked is Leeuwenhoek would put a sample and it would be held in position by this little turn screw device, and he would look with his eyeball through this little ball of glass at either a candle or the sun, and he could see objects in here. And if you made the ball small enough and perfect enough, you could get magnification up to 400x. So Leeuwenhoek wrote papers, and he sent them in Dutch to the Royal Society. And Hooke had them translated and then became so enamored with Leeuwenhoek that he actually learned Dutch so he could translate the papers himself. They had a lifelong collaboration, never met each other. So, over the years, people started to draw, and Leeuwenhoek coined this term, animalcules. I like this one. So they started to see shapes. They started to see behaviors. And the way they thought of this originally, these were microbes, but they thought of them as little animals with sex organs and stomachs and brains and eyes, but they were just miniaturized. It took a very long time for us to realize that they have a different behavior. And the behavior, I'm just going to go back very quickly to this slide, is something that is not obvious and was not at all obvious to Darwin, because Darwin didn't know what the word gene, there was no word gene yet. Gregor Mendel didn't exist, and the word gene was coined actually many years later. But the point is here that the distribution of function of genes or inheritance that Darwin recognized was descent with variation followed by selection. Descent with variation followed by selection. Here, many microbes can exchange genes laterally. No sex. I have a DNA piece. Somehow either I die or a virus takes my piece of DNA and gives it to you, or I secrete some piece of DNA into the environment and you can take it up. And there is no descent with variation. It's one of the major modes of evolution. Now, when we talk about the machines, I'm talking literally about nano machines. This is not, again, not an analogy, it's not a metaphor. So this Rube Goldberg apparatus is the apparatus that is responsible for splitting water that makes it possible for you to breathe oxygen. It has two major places where light is absorbed and is used to move electrons around. And this particular one has 4 manganese atoms in it. I'm going to show you this in a second. Now, here's a very interesting problem. There's nothing else like this on the planet. It was invented and came down to us, it descended to us once. So we don't know its evolutionary history. We don't know the other half of that bridge. We don't know how it evolved. We have its structure, and inside the structure, we have this from X-ray crystals. It's a very interesting and long story, and I'm not going to go into it, but it took 25 years for Horst Witt to crystallize this protein and to get the X-ray structure. And he died before he could win a Nobel Prize. So that Nobel Prize was never given to anybody yet. Here are the 4 manganese atoms. We know exactly where they are in the structure, but amazingly, we don't know exactly how it works. We do not understand how it works. So we have people around the country that are trying to mimic this with artificial catalysts. So Dan Nocera is very famous here at Harvard for this. Gary Brudvig at Yale, Chuck Dismukes at Rutgers, Nate Lewis at Caltech. There are a number of chemists. Why? Because if you could mimic that reaction, you could generate hydrogen. And if you could generate hydrogen from water, we fundamentally change the way we develop a fuel. You don't need fossil fuels anymore. So it evolved once in the prokaryotes, the organisms that are on the bottom part of that tree of life, in what's called cyanobacteria. And then those cyanobacteria were appropriated by other cells. This is a diatom. And these are the organelles called chloroplasts or plastids that are in every oxygen-producing organism on the planet. There's a dinoflagellate, this is a coccolithophore, and here is the distribution of the oxygen-producing organisms in what's called eukaryotes, and here's the only one of these in the prokaryotic lineage. So the only bacterium group that can split water and make oxygen are cyanobacteria. Everything else that's photosynthetic doesn't use water. Now, the spread of this across the planet, on land and in the oceans, created a vast machine that produced a lot of oxygen, splits lots of water, and this is a real seasonal map of the satellite images of the ocean and land. sped up, obviously. And the burial of the organic matter allowed now for you to breathe oxygen. Now, I'm just going to go through this. Let's talk a little bit about the burial. The burial occurs when the carbon is sinking, for example, in the ocean to shallow sediments and is then retained within the sediments, and over time is buried and buried and buried and buried by more and more and more sediments. And some of that is uplifted onto land, some of it is not uplifted onto land. But if you think about it, when you go and you fill up your car with gasoline, you're putting fossil diatoms or fossil algae, their remains, that's the fuel. You're using fossil sunshine. And every bond in your body, in my body, is derived from sunshine. Now, where do we bury the organic matter? We bury it on shallow seas, in continental margins. And this is why we have oil drilling rigs in the Gulf of Mexico, and we can have them off the coast of California, and we can have them in the North Sea, for example. We can also put that material up on land, and this is a very famous picture from the so-called Twozo-Wilson cycle, who was a geophysicist at the University of Toronto. And if we start out with this large block of thick but buoyant rock, granite, doesn't seem very buoyant, but it is buoyant relative to the ocean basalt, it floats, and we're standing on a piece of it now. Right? Over time, you put a heat engine under here, you can crack it, it can start to go away, and as it goes away, it's not like California is going to smack into Japan. Ultimately, we're going back to France, which may disappoint some people, but in any event, that's what's going to happen, but you won't be around to see it. That's another couple of 100 million years. When that happens, the material that is on either one side or the other that was in a shallow sea gets uplifted onto land and is stored there as the organic matter. And for example, here's a black shale from a very nice sequence in Spain we worked on. And you can see this is all the candy. This is candy. Okay? If we didn't have this process, we wouldn't have oxygen to breathe. You could make the oxygen, but it would just be consumed by those microbes. Now, over time, the metabolism of the microbes changed from these very, very, very strange metabolisms that happened very early in Earth's history to a place in time which is about 2.4 billion years, 2.35 billion years, which is we call euphemistically the Great Oxidation Event, where the burial of the organic matter exceeded the consumption of it by microbes, and oxygen was produced into the atmosphere for the very first time to a large number, to a number maybe between 1% and 2% of the present atmospheric values. And then we got the metabolisms that we see today. And later on, we got another burst of oxygen, late, about 750 million years ago, leading to the rise of multicellular animals. So microbes really made this Earth habitable. There were no plants. Plants didn't occur until 450, 500 million years ago. So for the first half of Earth's history, this was the ball. So you had perturbations, you got hit by meteorites, God knows what. The second half of Earth's history, the ball got out of that bucket and went into the new bucket of oxygen. And it never went back. It was a one-way street. Now, several years ago, this looks weird to you. Don't worry about it. It will not be on the test. A couple of colleagues and I, Tom Fenchel and Ed DeLong, who was then at MIT, now is at the University of Hawaii, not because he didn't get tenure at MIT, just that he was smart. We started to say, can we make a wiring diagram of the planet? And this is the wiring diagram of Earth. This is how organisms connect the products of their respiration to make a substrate for a new food for something else. It's literally a wiring diagram. It may look like this, but this is actually more complicated. Now, it turns out that there are only about 400 genes required to make that wiring diagram work. Very few genes. And I just want to show you that the distribution of those genes, they almost all contain transition metals, and iron is the most abundant of the transition metals in those proteins, followed by copper, a little bit of manganese, a little bit of nickel, a little bit of molybdenum. Now, I'm just going to have a little fun for the next 3 or 4 minutes with you because I think it's just fun. Many years ago, Joe Reed at Scripps Institute of Oceanography noticed that there were areas of the ocean that even in the summer when the light was bright, when the sun was out, there was high amounts of nutrient, in this case phosphate, but I could have plotted nitrate here. There's eastern equatorial Pacific, the subarctic Pacific, and the southern ocean altogether. These are called high nutrient, low chlorophyll conditions. So normally one would expect if you see nutrients like phosphate or nitrate or fixed ammonium, that they would be sucked down to nothing like most of the ocean is, because phytoplankton would just use them. The photosynthetic microbes would use them. But these 3 areas were anomalous. Several years later, the late John Martin, who was then at Moss Landing Marine Laboratories just north of Monterey, went out with very, 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 very clean techniques. And I'm not going to go through the whole detail, but he discovered that everybody that had measured trace elements in the ocean before contaminated them with their sampling gear. Everybody. And he very carefully measured the iron concentrations through the water column and the manganese concentrations. This is depth. This is 1,000 meters, 2,000 meters. I'm sorry, you don't see it so well. So the answer is here. The concentration of iron in the surface ocean of the Pacific, for example, is 10 to the minus 12th molar. Extraordinarily low. Very, very, very low. So he proposed that these high nutrient, low chlorophyll areas of the ocean were iron limited, which is interesting because if you need iron for all these proteins and you don't have iron, you're not doing very well. Where does the iron come from? Well, in the modern world, this would not have been true when you had no oxygen in the ocean. So for the first 2.5 billion years of Earth's history, iron was terribly abundant. That's why the proteins got the iron, because it was so easy to get. But then they bought into this material that went away. And there was a shortage of supply. Okay? So the iron now, for example, in the North Atlantic, is primarily derived from the wind-blown dust off of the southern portion of the Sahara Desert called the Sahel. So this is iron in the atmosphere. And if you are in these islands, this is Tenerife or Las Palmas, the Canary Islands, the sky is yellow. Darwin actually noticed this. The Voyage of the Beagle, Darwin noticed that the sky was yellow. He didn't really quite understand it, but it's fascinating that even he understood that this was a strange phenomenon. Now, if we take a look at the iron distributions of the ocean, they're much, much more abundant in the northern hemisphere than the southern hemisphere because there's much less land mass in the southern hemisphere. Now, this is the fun part. So, we participated, my group, in 11 iron fertilization experiments. So this is one of the later iron fertilization experiments. Here we're taking the mixing chamber out on the Melville. This is in Christchurch, New Zealand. This is a ship that is run by the Scripps Institute of Oceanography. We're heading out to sea. The guy who took the photographs was a friend of mine. He's a professional photographer. He has to be cute. So anyway, this is just now the planning session for how we're going to do the sampling. And now we're coming to putting the reagents into the test tube. So this is the reagent bottle, a little bigger than the ones we use in the lab. It's got iron sulfate in it, ferrous sulfate. opening up the test tube. Here's the mix master. There goes the iron. It's going to be mixed with a little bit of acid and then seawater to solubilize it. And this is a very sophisticated system for deploying the iron. It's a Costco hose with a lead weight. And there it goes off the fantail. And we turn it on, and we start delivering the iron. Very, very sophisticated. And I'm just going to leave this on for another few seconds while I talk over this. So we're delivering iron, and I'll show you how it goes, but it's like mowing a lawn. This is how we sample the ocean. These are called Niskin bottles, named after a Danish oceanographer who designed them originally. This is a rosette, and they will go down with the open top and bottom, and at a specific depth. There's an electrical circuit down to them on an electrical wire at a certain depth. They'll be triggered, and one bottle or two bottles will close at a specific depth, and you know the depth because there's a depth sensor on there. There are other sensors on this piece of gear. So that's the one that if you use that and you measured the iron, you're off by 2 orders of magnitude. This is the one that's been acid washed. There's no iron exposed here anywhere. And that's the one that's used for clean technique. That's the one that gets you the really precise iron concentrations. And this is what the real dream of a job of an oceanographer is. If you ever thought you wanted to be an oceanographer, you have to be able to do this ad nauseum. Take samples from a Niskin bottle, put it into a small plastic bottle, and filter it. That's it. Okay? And this is one of our early fluorometers. Okay, so this is what we do. We take the sample and we go back and forth and back and forth and back and forth like mowing a lawn. And you know the time you started, you know the time you ended. And then we cut across and go back and forth like this, okay? So we have these little triangular boxes here, these diamond-shaped. These are outside of the sample, right? So you have a control. Before you added the iron and after you added the iron, you have a control. And then we're going to go back and forth within this, and I'm going to measure how well that reaction center that splits the water works. And I'm not going to explain how you do that, but it's one of the things that made me sort of famous. It's what's called a variable fluorescence technique, and this is the result. So before you add the iron, this machine only operates at 25% efficiency. Within 1 day of adding the iron, the efficiency doubled. That nanomachine now clicks into action. So this is not an imaginary experiment. We did this many times. We can actually observe the machines functioning much, much better in these nutrient-poor waters when we add the iron. You increase the photosynthetic efficiency by a factor of 2. And ultimately, you make a patch that you can see from space, and ultimately, phytoplankton become food for zooplankton, and you increase the productivity of that region. Now, let me come back to the last portions of the talk. So I really think of the surface of the planet like a circuit board. I began as a kid. I didn't ever take a biology course until second year of college, I guess. I went to one of those quirky high schools in New York where I wanted to be an engineer, and they didn't teach biology unless you were really bad in science. So they only taught physics, chemistry, maths, and electronics, and stuff like that. So we have a power supply, the sun, one power supply, right? Now we need some wires. So I told you before, you're not making in this room, we don't have a big algal pot here making oxygen for you. You don't have to walk around with algae on your back. Some organisms do. So the atmosphere is a wire. That's one wire. The ocean is the second big wire. So the two big geophysical fluids on the planet connect all organisms to each other. So the air you breathe could have been made in Tokyo 100 years ago. And you're breathing it now. So this connector, those yellow and blue and red lines are really 1 or 2 wires. We have a power supply, and now we have to figure out what the organisms do. Sorry. So these little processes here, you and I are acting like basically a transistor. And I'm not going to go into the electronics of a transistor, but I just want you to understand that inside of us, we have these iron-containing proteins. So this is a 4 iron and 4 sulfur protein. This is a very easy mineral to identify. So there are many 4 iron, 4 sulfur cluster proteins that contain this motif, and it's all bound on the same amino acid motif around it. So that's for iron. Now we can elaborate on this. We can make some really, really fancy molecules. So this is the iron-sulfur complex. There are now 38 irons in this protein, and 1 little molybdenum sitting right there. That's the only protein on the planet that fixes nitrogen, takes nitrogen from the air and converts it to ammonium, without which there would be no life. So guess what? It's a really, really, really important protein. We spend 23% of the energy of agriculture in the Haber-Bosch reaction, taking nitrogen from the air to do this at high temperature and pressures with hydrogen that we derive from coal. By the by, for those of you who have never seen the Haber-Bosch reaction, if you're ever in Berlin, in the Holocaust Museum in Berlin, Fritz Haber's original machine, his catalytic machine, was there. It is there. It's a box about this big. It could produce about 1/2 of a glass of ammonium in an hour. Carl Bosch realized the potential of this. He got BASF to build a plant in Germany to make ammonium. It was the first massive producing ammonium plant ever in the world, obviously. Hitler came to power. He said, I don't care about your stupid ammonium. I want nitrate. I want nitrate so we never run out of nitrate for munitions. So Bosch was horrified. But he created a process that produced ammonium, and he took the ammonium and he made ammonium nitrate, which is the common form of the fertilizer today. This is what Timothy McVeigh bought. It's explosive, but that's the reason we have it, is because of this accident of history. These guys don't make any nitrate. They just make pure ammonium. So if we could figure out how to do this reaction, again, water splitting, here's another one, nitrogenase, we would save an awful lot of energy on the planet. Here's another little enzyme. This is an enzyme in you and me that converts those nasty reactive oxygen species into something very benign. So I don't know how many of you run or do really aerobic exercises. Don't. You're killing yourself. You only have 1.5 billion heartbeats in your life. You can spend them any way you want. But by breathing oxygen very rapidly, you create these nasty molecules, and you have to employ a lot of this stuff to get rid of them. Better not have to do that. And we have many of these. Oxygen is a pretty toxic molecule. So if I go look at the structures of those 400 proteins that are responsible for moving the electrons around, there are actually only, we see, about 35 motifs. So let's take a look at the parts list now. The parts list: 1 power supply, two wires, 35 transistors in the box. Shouldn't be too hard to recreate the other half of the bridge that we didn't see. Shouldn't be too, I'm going there, I'm trying. Now, this is the evolutionary history of those enzymes, the 400 enzymes that create life. So we start out here with this 4 iron, 4 sulfur cluster, and it was a hydrogenase. It used hydrogen as a mechanism for converting carbon dioxide to sugar. Bugs wanted to make sugar very early. Okay? And here, way, way down here, is the cytochrome C oxidase. It contains copper. That's the enzyme that allows you to breathe oxygen. So this is one of the latest evolving enzymes, something that allows you to use that waste gas that was produced by the cyanobacteria. Now, I'm going to finish this up in a fun way because this is where I've been for several years. So in the Archean world formally starts somewhere around 4 billion years ago. It wasn't on a Tuesday, but it was like that. And then it ends up at about 2.5 billion. In the Archean ocean, or the Archean atmosphere, there was no ozone because we didn't have oxygen. So this is the sunshine that comes from our star to the top of our atmosphere. And these spikes are not noise. These are lines of absorption of elements on the surface of our star. So the most famous of these lines of absorption are for iron, for example, the Fraunhofer lines. But Lyman himself, who used to be here, and he named his sons. Can you imagine this? He has a son named Harvard Lyman, so we call him Harvey. But he had another son which was called Princeton Lyman. He had a son called Yale Lyman. Oy. In any event, this is the distribution of our star energy, our star's energy at the surface of the Earth. And before we had any ozone, this energy would have hit the surface of the planet. This is ultraviolet radiation. No problem. Now, this is an experiment we did. One of my really, really smart students was a chemistry student, just pure chemistry. So we make this molecule here, which is iron carbonate. So you're familiar with calcium carbonate, which is chalk, or magnesium carbonate, which is something that you might take to make your stomach feel better if you're not feeling too good. This is iron carbonate. It's FeCO3. It's not very stable in oxygen world, but it was a really important element or mineral in the early Earth. It's called siderite. Now, for this demonstration, I don't know if you can see this, but we expose the siderite to ultraviolet radiation. And you see it turns orange? We popped with photons, with light, an electron out of the iron and created rust. No biology. Now, the product, if you're a chemist, you pop an electron out of something, it has to go somewhere. There's no such thing as a free electron, except in a laser. It goes and it produces hydrogen gas. Amazing. So you have a very, very simple, non-biological reaction that produces hydrogen gas early in Earth's history. At least 2 processes now. The photooxidation of siderite as a mineral, for example, or volcanoes in mid-ocean ridges, and volcanoes in general. Now, what's wrong with this picture? This reaction is not catalytic, it's sacrificial. So if I do it and I use up all of that iron, it doesn't come back. I just did it once, one-way street. Whereas if I put a microbe in here and I shine the light on and it produces oxygen, it doesn't use up any of its stuff. The manganese comes around and around and around and around and around and again, and it makes the oxygen. So how do we go from this reaction to something that's catalytic? You know the answer? Nobody knows. That's on the other side of the bridge. And I'm going to talk about that tomorrow because it's a really important thing to think about technically. But this is another very simple reaction. We're taking, in this case, manganese carbonate. We're exposing it to ultraviolet radiation. You can see we oxidize the manganese. And now what do we produce? We produce formic acid. This is like candy. No biology. So we're doing abiological reactions with light. I'm trying to get to very, very simple Very simple catalysts. And I'm going to end up with this because I'm sitting on an astrobiology working group for NASA, which is going to be leading, at least informing, the so-called next decadal review. In the last decadal review, we had this terrestrial planet finder, which was killed in the decadal review because it was too risky. It had two components, what's called a chronograph and an interferometer, and both of these would have flown at the Lagrangian 2 point, which is the equal gravitational distance between the Earth and the Moon. I'm not going to bother you with the details, but the idea was to look at the dark side of a planet as it crosses in front of its star and to detect the gases on that planet. So if you, for example, had high concentrations of ozone or oxygen and methane, you could potentially ascribe that to life, even though you'll never get there, because these planets are very, very far outside of the possibility of getting to them, at least with any conventional technology that we have. So this is what we do now. We do what's called limb experiments, where the small amount of gas that is in the atmosphere on the surface of a planet as it transits the star is lit up from behind by the star, and we try to observe the planetary composition this way. It's not very efficient, and it's got a lot of noise in it, but at some point in the next decade, I am hopeful that with the number of targets that we have, there are about 3,800 extrasolar planets that we've discovered. Most of them are gaseous and too close to the star, or mostly too close to the star to be habitable. But some point we will see at least terrestrial planets, rocky planets, in front of sunlight stars. And at that point, we will have a big question to address and answer if we can detect the gas composition. And that is, are we alone? So did the bridge start from one place, or could it have started from many places? So where did we come from and are we alone are the two mantras that NASA has followed for many years, and I'm very proud to have been part of that, at least, effort to frame them as questions with missions. So I'm going to just wrap this up. I just want to say the first 2.5 billion years of Earth's history was the research and development phase. This was the Silicon Valley of the solar system. Okay, and we got people like, Bill Gates and so on that created these microbes. Now, once they locked into that machinery, it's just exactly like Microsoft. So rather than reinvent an operating system, you made another 500,000 lines of code to correct for all the mistakes that you had in the original code. And the machinery became clunkier and clunkier and clunkier, but if it worked, you just keep using it, okay? And so we are left with these key metabolic processes that are all developed in these prokaryotic organisms, bacteria and archaea. So as one eukaryote talking to another, very frankly, we're very boring from a metabolic perspective. We don't do very much interesting. We're not really different from Escherichia coli, except we have brains. So we're the animalcules. They're the originators. Just putting it in perspective. There are only about 400 core metabolic genes that make the world go round, and these metabolic sequences are coupled on basically the entire globe. So the entire process, I just want you to appreciate this because it's really, really interesting when you think about what life could be on other planets or other planetary bodies like Europa if it's not directly driven by light. So I'm going to say thank you very much for your attention, and I'll be happy to answer any questions you might have.